Water pollution is a common environmental problem in the world today. Often we think of water pollution as caused by human activities, as was the case for the recent water crisis in Flint, Michigan. But is all water pollution caused by humans? Not always. For example, groundwater contamination by arsenic is a natural occurring process and is becoming a worldwide environmental issue. In South and Southeast Asia alone, more than 100 million people are exposed to arsenic-contaminated groundwater, leading to severe health problems including cancer and skin disease.
Arsenic is a chemical element that is often found naturally in minerals. If you drop a rock containing arsenic into a cup of pure water, odds are not much will happen because most forms of the mineral are not soluble in water alone. However, if we add the right mix of microbial organisms and organic carbon to this cup and take out the oxygen, the mineral will break down and release arsenic into the water. Even though all this is happening naturally, human activities such as well drilling and irrigation can change the groundwater flow pattern and introduce more organic carbon that will further facilitate the arsenic release process. If your well water turns out to be contaminated, there are a few options to regain potable water in your household. The first option is to join a public water supply system. The second option is to install an arsenic filter for your well. The last option is to drill or switch to a new well that is more likely to produce arsenic-free groundwater. In less developed countries, the last option is often preferred due to the absence of a public water supply system or effective water monitoring and treatment equipment.
Today, international science communities such as MIT have built many study sites in arsenic-affected regions. The research effort has also motivated programs that are in place to help affected populations regain clean drinking water.
所以这个是刚刚跟大家提到的这个在饮用水的部分。没有需要把字幕转成是中文的吗？还是看英文的字幕还可以？需要转成中，但是它的翻译可能会有一点失真，有需要吗？好。好，那这个就是刚刚讲，那个 MIT 的那个教授 Charles Harvey 他给的谈话，然后再讲孟加贝格那边的问题。那他这边会着重在就是道理，但是他一开始会先给大家一个就是整个 background 的介绍。
Bangladesh is situated on the river deltas and floodplains of the Ganges, Brahmaputra, and Meghna rivers. It is a densely populated country. Bangladesh was devastated in the separation from Pakistan in 1972 and then experienced severe food shortages, disease, and natural disasters. Over the last decades, it has been rapidly developing, and the population is now stabilizing with a remarkable density of over 1,000 people per square mile. Arsenic contamination of well water in Bangladesh is often called the largest mass poisoning in human history. Our research group from the Bangladesh University of Engineering and Technology, MIT, and Columbia University has worked together to understand this problem. Over the last decades, most Bangladeshis switched from drinking water from ponds and rivers to drinking groundwater from wells. They switched because the pond and river water was contaminated by disease. Tragically, the groundwater is contaminated by arsenic, 10 to 100 times higher than safe. As a consequence of this switch in water supply, millions of Bangladeshis experience arsenic poisoning that causes cancer and heart disease. Arsenic poisons plants in similar ways as animals, and we are currently working on how arsenic in irrigation water affects rice fields. Rice is central to most everything in Bangladesh. It is part of nearly every meal and is the primary source of calories for people. Rice fields cover much of the landscape, accounting for 75% of agricultural land. Village life is strongly tied to the seasonal cycle of planting and harvesting rice. Bangladesh rice is grown by individual farmers who cultivate their own fields. Rice is grown in standing water. In the summer and fall, monsoon water floods rice fields. In the dry winter and spring, rice is irrigated by groundwater. Traditionally, rice was grown during the monsoon, under natural monsoon floodwaters in June. This rice is called almond rice, and some can grow in very deep water, up to several meters. Now a second crop, boro rice, is grown during the dry season, from December to May. It is dry and hot during this season, and pumping groundwater to irrigate provides the standing water in rice fields. Dry season boro rice is now the most important crop in Bangladesh, and some farmers no longer cultivate almond rice, particularly where floodwaters are very deep. Irrigated boro rice has greatly increased rice production, enabling Bangladesh to be largely self-sufficient and even export some rice. The groundwater used to irrigate rice is contaminated by arsenic that is naturally present underground. This arsenic, carried by the irrigation water, is then deposited in rice fields and accumulates in the soils over years. Our team of researchers is studying how rice yields are affected by arsenic and what can be done to fix the problem. We found that rice production is reduced by as much as 20% by the buildup of arsenic in the soil from irrigation water. To figure this out, we excavated soil and moved it between small plots in 20 different rice fields, some fields with arsenic contamination and some without. We weigh and record the rice harvested from all these plots. In this way, we compare the rice field between soil with and without arsenic contamination, so that we can determine how much rice yield is damaged by arsenic buildup. We have also shown that the problem can be temporarily fixed by replacing the top 10 centimeters of soil with uncontaminated soil. The problem of providing uncontaminated water, both for irrigation and more importantly for drinking, remains largely unsolved. Bangladesh is unlikely to have uniformly safe water until it has piped water from centralized treatment systems, much like the rest of the world does.
好，所以这个是 Professor Harvey 对于 Bangladesh 那边所做的介绍，这样。然后我们接下来要看的是另外一个在 Stanford 也是专门在做生的一个老师，叫 Scott Fendo。他会去讲到，就是现在这个生跟稻米之间对稻米之间的冲击，但是会连接到 climate change， 一样就是让它自动翻译。那最后一个我们要看的，就是Harvey 的那一个团队，经过10年，最后找到为什么就是他们那边地下水高浓度生的那个学生，一个女学生叫 Rebecca Newman 这样。那 Newman 现在在 University of Washington 在Seattle那边任教。那他开始任教之后，也有持续在做生相关的研究这样。他待会也会去提到他的实验室现在在探讨就是生跟稻米之间的问题这样。我们先看那个先看史丹佛的这个code 这样。那我要讲的是，你可以看到，就是说这些其实都是环工的老师或是环科，那他们的研究不会是只有就是在实验室的尺度这样，就是说你要去做到 field work， 其实相对而言挑战性会比较大，因为我们一直在讲，就是说环境整个系统是很复杂，所以你要把你调查的结果去理出一个脉络出来，这个是要有很大的功力跟挑战才可以做得到。
Rice is the largest staple crop in the world, and one of the big challenges as we go forward is that temperature is really going to stress rice out. The IPCC's mid-range evaluation is that we could be as high as 5 degrees above what current temperatures are. And what that does to rice is it stresses the rice and you get a yield decrease of around 10 to 12% of current varieties right now. What's missing in that is that most of the rice growing regions have a fair amount of arsenic, and arsenic concentrations in the soil that are increasing due to irrigation with arsenic-contaminated groundwater. When you take the elevated temperature and soil arsenic concentrations in collectively, you get about a 40% reduction in yield. So think about that. If more than 50% of the world's population is dependent on rice, and you're cutting back that food supply by 40%, that is a huge hit. There's a variety of different ways that you can try to emulate climate change. What we chose is a greenhouse study where we can really tightly control the temperature and also tightly control the CO2 concentrations. We use soils that were from California, but we've also done experiments where we have soils from Bangladesh or from Cambodia, and we grow the rice within pots within the greenhouse.
It's a multifactorial experiment design, so we have two temperatures, that is ambient temperature and another elevated temperature by 5 degrees Celsius. Another factor that we are returning is the atmospheric CO2 concentration. So we also have two. One is the control, and another one we double it. And then we monitor the growth of a plant. We take soil data. We monitor plant physiology data. Basically means that the height of the plant, how many tillers and panicles they developed, and then we harvest them in the end. And after harvest, we digest them with the leaves and grains and all the plant tissues there, and trying to analyze how much arsenic and consequently other micronutrients that's in there.
The experiments themselves take about 4 or 5 months to run, but we've been doing these experiments over and over again, so they've actually been running now for 3 or 4 years. We repeat these to make sure that we have replicable results, and also that we can see it with different soils and slightly different varieties of rice. This is a wake-up call of what we're going to see if we don't do something about greenhouse gas production. You can think holistically about coming up with new varieties of rice that are going to both be able to be temperature tolerant, but also arsenic tolerant. I'm also optimistic that when people start shining a light on the threats that we see at a 5 degrees Celsius impact, that we won't get there, that people will wake up that that's just unacceptable. That's my biggest hope and my biggest room for optimism.
好，所以就看大家有没有看待这件事情的时候，有像他一样那么乐观在看待这件事情。我们就要看的就是我刚刚讲的，就是Newman 他就是开始当老师之后，他在西雅图那边所做的一些相关的研究这样，所以他整个research site 从 Bangladesh 转移到 Seattle 那边，然后我们会从他讲到做跟讲那个在他们那边的湖泊Fish South 那边的状态，因为那个地下水那边就是算他以前当学生的研究，所以比较不是新的东西。
Okay, so Bangladesh, West Bengal raised a lot of awareness about the possibility of arsenic in groundwater and really motivated a lot of research into understanding how arsenic behaves. At this time, though, we also started to think about the possibility of arsenic making its way into the food system. And this is because in Bangladesh, as I mentioned, groundwater is the primary source also of irrigation water. So farmers in this part of the world, they grow a dry season rice crop and it gets irrigated with groundwater, but this groundwater has arsenic in it. So this means that every year when the farmers are irrigating their fields, they're adding a little bit more of arsenic onto that soil every year. And so researchers started to wonder, well, what does that mean? What does it mean if we're adding arsenic and increasing arsenic concentrations in our soils? So in general, this is one of many studies coming out of Bangladesh. In general, when arsenic in soil increases, we tend to see increasing concentrations in the grain of the rice plants grown within that soil. This is not always the case, but most of the time this is what we see. And then what was more surprising, a publication coming out around the same time, is that we actually saw a decrease in grain yield. So as soil arsenic is increasing, we're seeing a drop in the quantity of rice which is being grown. So not only is arsenic bad for the human body, it's also bad for the rice plants. So basically the rice plants are struggling when they get exposed to arsenic, and they're just not as healthy, and so they're not producing as much rice grain. So evidence from Bangladesh that this contamination of soils with arsenic negatively affects both the quality and the quantity of rice which we're growing. So this was focused on Bangladesh. Researchers started thinking, well, what about the rest of the world? Rice is grown in more places than just in Bangladesh and West Bengal. And so these researchers, they went around and bought polished white rice from a number of different marketplaces all around the world, and they looked at the arsenic content of that rice which they collected. Here I'm showing you data from their study just for Bangladesh in the United States. They went to a lot of different places. And this top panel is the model global distribution, so a synthesis kind of all the data that they collected from all the different countries. Okay, well, surprising. Rice grown in the United States has more arsenic in it than rice grown in Bangladesh, right? So I just was telling you, well, they're irrigating their fields with arsenic-contaminated groundwater, and yet we're growing rice in the United States that has more arsenic in it than the rice which is grown in Bangladesh. It turns out that in the United States, in the southeastern part of the US where we grow most of our rice, we're growing on former cotton fields, and we used to use arsenic-based herbicides on these cotton fields. So this is an example of that legacy arsenic contamination coming back and having modern-day consequences for our food system. Okay, so what do these levels of arsenic in rice mean? So unfortunately, there is no standard for arsenic in food. So in drinking water, we have a drinking water limit. 10 micrograms per liter is the standard that's currently set here in the United States. If things are above that, it's considered unsafe, and if we're below that, it's considered safe. Food doesn't have anything like that, so the best we can do is kind of deal with rough guidelines and thresholds based on results from academic studies. So in 2013, another pretty foundational study came out. This was the first one to connect arsenic in rice directly with a human health effect. So it's always that kind of direct connection, I think, which is hard to make when we're trying to say X causes Y. So this is one of the first studies that was able to do that for rice arsenic. And what they showed is that when you exceeded a concentration of 0.2 milligrams per kilogram of arsenic within the rice, you started seeing genotoxic effects. So that basically means that the DNA got damaged. And when we have damaged DNA, we get mutations to our cells, and then cell mutations can lead to cancer. So it's basically like a first step on the road to cancer. So that threshold is at 0.2 milligrams per kilogram. And then more recently, in 2016, the Food and Drug Administration finally came out with a guideline, but it's pretty narrow. So this is regulating, or it's not even regulating, it's just a guideline for infant rice cereals. Okay, so a pretty narrow set of things that we're thinking about, but nonetheless, it's a guideline, and they're saying we should not exceed 0.1 milligram per kilogram of arsenic in infant rice cereals. So when we look at these kind of thresholds, we see, well, much of the rice in the world is below this genotoxic threshold. A lot of it is, at or slightly above this guideline. Of course, in the United States, our rice is well above this genotoxic threshold. So work that I've been doing here at the University of Washington has been thinking about how is this issue of arsenic contamination of rice going to change as we move into the future. And we have an altered climate system. So here on this map, I'm showing average regional rice output. Not surprisingly, most of the rice in the world is being grown in South and Southeast Asia. And then next to this, I'm showing predicted changes in average surface temperature. This is resulting from the most recent IPCC report. This top panel is kind of the best case scenario and this lower panel is our worst case scenario. And what this is showing us is that in these rice growing regions, we can expect surface temperatures to increase anywhere from around 1, up to 5 degrees Celsius. So we're moving into a warmer future. Well, why should we care about warmer temperatures? Microbial reactions, they get faster when temperatures get warmer. So the hypothesis that we had when we moved into this research project was that under warmer temperatures, this microbial reaction is going to speed up, and so we're going to get more movement of arsenic off of the rice soils and into the soil water. So if we have more arsenic in the soil water, this means we can get more arsenic being taken up by the roots of the plants, more translocation of that arsenic into the above ground biomass and into the grain of the rice plants. So to test this hypothesis, a graduate student in my group, Yasmin Farhat, along with the help of multiple different undergraduate researchers, conducted an experiment. So she grew rice plants under 4 different temperatures. And then once they came to maturity and the grains had fully ripened, she basically collected and harvested these rice plants and analyzed the arsenic content in the tissues of the plants. And these data are weeks old, so she worked so hard to get these data to me in time so I could show them to you tonight. So this is exciting. What I'm showing you is average stem arsenic concentrations and then average grain arsenic concentrations as a function of the temperature at which the plants were grown. And so what we can see is a very clear, strong, positive correlation between levels of arsenic in these above ground tissues and the temperatures at which the plants were growing. You might be noticing that we seem to be missing this top data point, so grain from the plant grown under our warmest temperature. Well, that plant was so heat stressed, it just didn't produce any grain. So the yield was 0, there was nothing there for us to analyze. We can compare our measured levels to these thresholds and guidelines, which I presented previously. We're working here with a pretty low arsenic system, so our soils were not contaminated with arsenic. So overall, we had a pretty arsenic-free system. So down in these lower temperatures, the grain that we were producing was below this FDA guideline, well below this genotoxic threshold. But nonetheless, what we're seeing, right, is even when we have a low arsenic system, if we start marching the temperature up, we can start moving concentrations up into these regions where we begin to start having some concerns. So unfortunately, the results of our experiment are suggesting that this issue of arsenic contamination in rice is going to get exacerbated as we move into warmer climate conditions. Okay, lots of bad news. You might be starting to worry about all the rice that you ate in the past, wondering if you'll ever eat rice again. I want to point you towards at least some concrete advice to help you deal with maybe some of that nervous feelings you have. So Consumer Reports came out in 2012 and 2014 with really 2 nice studies. They did a comprehensive review of arsenic in rice and a range of rice-based products, and they've also started to look at other grains like quinoa, for example. And in their reports, they provide some really good take-home messages for consumers. One of them is they identify the types of rice that are low in arsenic. So these include basmati rice, grown in India, Pakistan, and California, and then sushi rice grown in the United States. So next time you're at the grocery store, you want to buy rice, focus on these varieties. They're the ones with the lowest levels of arsenic. They also highlight the rice that has the most arsenic in it. So avoid, if possible, rice from Texas, Louisiana, and Arkansas. So these regions where we have grown rice on fields where arsenic-based pesticides were used in the past. And then in general, regardless of the variety, brown rice has more arsenic in it than white rice. And this is because the arsenic actually gets deposited on the outer part of the rice grain, so the part that makes the rice brown. And so when they polish that rice and make it into white rice, that outer sheath gets taken off. So it brings down the levels of arsenic. So if you have a choice between brown rice and white rice, go for the white rice. Okay. I know it's opposite of what you maybe thought. You thought, oh, brown rice is so healthy, I'm getting whole grains. But and then I think a really, really nice outcome of the report is that they actually developed a point system. So they say aim for no more than 7 points a week, and then they give you this table where they outline, you know, for different rice-based products, for a given serving, how many points is that for a child versus an adult, okay? And this is just a snippet of their table. It's quite long and extensive. So I would say, you know, if you like to eat rice, if you're gluten-free and you just inevitably end up eating a lot of rice, this would be a good table to kind of look at and gauge and try to just make sure you're staying in safe levels. Okay, so we've been talking about arsenic in water, groundwater, arsenic in our food system. We've been thinking kind of globally and South and Southeast Asia. So what I want to do now is bring the discussion local and talk about arsenic in aquatic ecosystems. So it turns out that here in the Puget Sound region, our landscape is contaminated with arsenic, and this is because the Esarco smelter operated for just under 100 years outside of Tacoma, Washington. And this was a copper smelter. Arsenic got released out of the smokestack just as an unwanted byproduct. It blew across the landscape, settled down, entered into our soils and into the sediments of our lakes. Washington Department of Ecology has a really nice program which is called Dirt Alert. And this is a snapshot just of the website. And here they show you measured concentrations of arsenic in soils throughout the region. So darker brown color is higher levels of arsenic, and then you move out into lower levels of arsenic. And the nice thing is you can actually go to this website and type your address in. So if you're worried about levels of arsenic in the soils around your home, you can type your address in and it will take you to that location. So I've just picked some place in Gig Harbor, which is kind of in the heart of the plume, and they can tell you, you know, what the expected concentration of arsenic is in soil near your property. They let you know if your property is inside or outside of the smelter plume. And then they also give advice on simple healthy actions you can take to reduce your arsenic exposure from this contaminated soil. So again, this is soil, so you're mostly just worried about that soil making it into your mouth. So they say wash your hands, keep your shoes outside of your house, mop up your floors, and wash off toys that maybe have dirt on it. So some real kind of easy, straightforward ways to limit your exposure to the contaminated soils. The Dirt Alert program is really great, but it's only focused on dirt. So an area that's been largely overlooked are the lakes in our region. So we know that the sediments of our lakes have also received this arsenic contamination from the Esarco smelter. My colleague Jim Gowell down at UW Tacoma, he went around to a bunch of lakes here in the Puget Sound lowland, collected sediment out of the lakes and measured the arsenic content. And those are the data I'm showing you here. Each of these dots is one of the studied lakes, and the size of the circle lets you know how much arsenic was in that lake. So the bigger the circle, the more arsenic. So we see that there are lakes all around this region which are contaminated with arsenic, and some of them extremely contaminated with arsenic. In general, I would say that there is a lack of understanding of how arsenic in these lake systems affects human and ecosystem health. And so that's a gap that I and a whole number of other researchers have been trying to fill. We're on a large collaborative project trying to understand arsenic mobility, bioaccumulation, ecotoxicity within these lake systems. There's lots of people in this project. I'm showing you just the people who are involved in the data that I'm showing you tonight. So there's a team of us in the College of Engineering. I've lumped my dad in as an honorary member. He came into the lab and taught me how to dissect fish so that I have fish data to show you tonight. So we can thank him for that. We have colleagues over in the College of the Environment and a number of people down here at UW Tacoma. Okay, so when we're thinking about arsenic in lakes, the story is very similar to the story that I've been telling all night long. We have arsenic in these sediments, it's on the solid phase, but we have microbes out there in the sediment. Those microbes mobilize that arsenic off of the sediment and into the surrounding water within the lake bed. Once we have arsenic in the water of the sediments, it can get transported up into the overlying lake water. Once we have arsenic in the overlying lake water, it can then get taken up into phytoplankton. So phytoplankton mistakenly take arsenic up out of the water. They're really trying to get phosphorus out of the water. It's one of their needed nutrients, but phosphorus and arsenic are chemically very similar. So they take it up by mistake, thinking it's something they need, and instead it's something that's bad for them. The zooplankton eat the phytoplankton, get exposed to arsenic, and then the fish eat the zooplankton. Of course, once we have arsenic in fish, we have the possibility of arsenic making its way into humans. So we've been studying these lakes for a number of years, and we've had some pretty surprising results. Notably, arsenic that we're finding in this aquatic food web is not directly related to the amount of arsenic that we're seeing in the lake bed sediments, or even the maximum concentrations of arsenic that we're finding in the overlying lake water. So what I want to do now is step you through our results and help explain why we're seeing this pretty surprising and unintuitive result. So we're going to focus on two lakes, Angle Lake and Lake Killarney. These are two of the most impacted lakes in the region. They each have around 200 milligrams per kilogram of arsenic within their sediment, so severely contaminated. When we look at the maximum levels of arsenic making its way into the overlying lake water, we find that both of them have very similar levels of arsenic, around 50 micrograms per liter, and these are maximum concentrations. But when we look at the phytoplankton, we see a huge difference in the concentrations of arsenic in the phytoplankton between these two lakes. So I want you to note this is on a log scale, so maybe visually it doesn't look that different, but it is quite different, and this is why I'm giving you the actual numbers here. Okay, so same sediment arsenic, same maximum water arsenic, but dramatically different amounts of arsenic being taken up by the phytoplankton. What the heck is happening? Well, it turns out that Angle Lake is a deep lake and Lake Killarney is a shallow lake. So why does this matter? It turns out that in Angle Lake, these deep lakes, these bottom waters often become stagnant and anaerobic. So this means that there's no longer oxygen inside of them. Aquatic life needs oxygen to survive, so that means that the aquatic life is hanging out at the top of the lake, and the arsenic is getting mobilized down here at the bottom of the lake. Okay, so we get spatial separation between where the arsenic is and where the aquatic life resides in these deep lakes. In the shallow lake, we don't have these deep bottom waters that become anaerobic. So when we're getting arsenic mobilized up out of the sediments, it's actually coinciding with waters that have oxygen in it, and so we actually get overlap between the contamination and the aquatic life. So we were thinking, well, maybe it's not maximum levels of arsenic that we should be looking at, but instead we should be looking at arsenic within oxygenated waters. So here I'm showing you data from 4 different study lakes, and I'm plotting concentrations measured in phytoplankton against the average concentration of arsenic measured in waters that contained oxygen. The circles are snapshots in time, so every time we went out to a lake, collected a sample of phytoplankton and collected our water samples. And then the diamonds are showing the average for the summer season. And so what we see is that arsenic bioavailability or uptake of arsenic into phytoplankton is related to the amount of arsenic that we're seeing in oxic waters. And this is noteworthy because water quality guidelines are currently focused just on these maximum levels of arsenic. But what our data are suggesting is that we should maybe not be looking at maximum levels when we're thinking about water quality guidelines. These guidelines are set to protect the health of the aquatic ecosystem. But instead of thinking about maximums, we should be thinking about levels of arsenic in waters that have oxygen, because these are the waters that are going to be housing the aquatic organisms that we're trying to protect. Okay, so if we come back now to our 2 study lakes, Angle Lake and Lake Killarney, instead of maximum levels, I'm now plotting levels of arsenic within oxic waters. And so you can see that we have almost an order of magnitude difference in the average concentration of arsenic within oxic waters of these 2 lakes. So now, when I throw up the phytoplankton data, which you've already seen, it makes a little bit more sense, right? We're getting now this increased transfer of arsenic from this oxic water arsenic into the phytoplankton, right?
好，所以到这边就好。所以呢，你可以发现就是说，他在做他的研究的调查的话呢，他们就是要去探讨背后的成因。所以我们先讲，就是说以他在湖泊的这个食物链所测得的这个砷含量的话，它就会连接到我们就是这堂课应该讲，就是生物有效性还有availability的这个事情，这样就是说你不能从表象去看，因为表象有可能给你一个就是错估的现象，那所以你要去探究事情背后的成因，这样，所以他们最后有去做了一些修正，就是从所谓的availability生物有效性的这个角度切入之后，才比较可以去解释他们的数据，这样这是第一点。那第二点，我希望大家有注意到的呢，就是他在讲他做的那个稻米跟砷之间的关联的研究的时候呢，他们都会有一个 hypothesis， 然后有这个 hypothesis 之后呢，然后他们去验证，然后看他们得到的结果有没有办法去支持或是支撑他原本的假说这样子。那这个就会呼应到我们一刚才所讲，这其实就是科学的方法，就是说你在做研究的话呢，你要用科学的方法，所以科学的方法是它有一些既定的程序，你要先观察到什么，然后你去鉴定，你去 identify 那个问题是什么。那接下来你要去 address 这个问题的话呢，你要去提出你的 hypothesis， 然后之后你要去做实验、做调查，去 check 这样的 hypothesis 是不是正确。那所以其实它就有就是他们做研究的话是遵循这样子的逻辑思考去做他们的调查，这样。这第二点。第三点呢，就是希望就是说我们这门课毕竟还是英语授课，所以我假如没有讲英语的话，那大家还是可以从这一些影片当中去多少练习听力之外呢，大家也可以从中去学习，就是说他们怎么做报告，就是说他比如说投影片怎么做，然后他怎么讲他的故事这样，因为这都是可以趁这个机会去学习到的东西这样。好，所以呢就看完生了的话呢，那其实大家可以回头想，就是说那在台湾的状况会是如何嘛？那刚刚也提到说，以稻米来说的话，他发现就是说，在美国本土的稻米种出来的这些应该说美国的谷物，美国的土地种出来的谷物呢，里面所含的砷好像都会比在亚洲或其他地区还要来得高这样子。那他特别去，然后后来 美国的 FDA 就针对了婴儿食用的这些米饼或是米类的制品去做规范。那这个重要原因是因为有些人可能对于就是麸质，就是一些麦类的某个成分会过敏，所以他只能吃米类的东西这样。那所以这个是生的问题，那所以就要回到就是说，我们亚洲人吃了很多的米，就是所谓的大米。那小米可能比较少人去研究，所以假如说你对这个议题有兴趣的话，搞不好你就可以思考从小米的角度切入的。那以大米来说的话，一样你会跟米的品种会有差异这样子。好，所以这个是生跟米的问题。那事实上，我们今天应该没有时间讲。其实汞，我们以往的观念都觉得说，它其实主要是因为水生食物链的关系，所以会有高浓度的汞。事实上呢，汞也会容易累积在稻作里面，而且会累积的其实是最毒的汞，甲基汞。我们刚刚一直强调，就是说这些微量元素、这些重金属，它的价态、它的 chemistry 不一样的话呢，它在环境所表现出来的 behavior， 它的行为，所谓的行为就包含它的移动性跟它所带来的毒性冲击都会不一样。那不同的元素的话呢，它会有就是它独特的化学性质，所以你不能就是一概而论，你不能说我在砷这边看到这样，那我就可以类推到其他的重金属，这是绝对不可能的事情。这样，那以砷来说的话呢，它的化学趋势刚好跟汞不一样。汞我们刚刚有讲，我们应该下礼拜讲，它其实是有机形态的汞远比无机形态的汞还要低。可是砷刚好是整个颠倒，它反而是有机形态的砷比无机形态的砷还要不毒许多这样。虽然都是毒性物质，可是以程度上来说的话，无机砷会比有机砷还要来得毒这样。那汞的话呢，也会在米粒里面去累积，而且会累积的是甲基汞。那当大家有注意到他所提的，就是说他们注意到那些米。只要有测到砷，其实主要是在麸皮，就是说，对他的建议说你要吃白米，你不要吃糙米，因为你吃糙米的话呢，那个麸皮会有砷的累积这样。那这个其实从营养学的观点来看的话，会是比较不好，因为我们知道白米其实是精制米，所以它其实没有什么所谓的有益的矿物质或是微量元素在里面，所以。营养学家都会建议你要去吃糙米，因为你这样子从营养的角度来看的话，会是比较就是好的一个状态的。那可是汞就不一样，汞的话呢，你会进到米粒的汞，刚刚讲是甲基汞，而且呢，它会穿越麸皮的这一个屏障，所以反而你在白米里面会测到的汞的占比不会输给在麸皮，那主要还是甲基汞。那汞的话，刚刚有讲，它最毒的就是有机形态的汞这样。好，所以呢，这个是汞的问题的话，我们就下礼拜再跟大家做介绍。大家这礼拜有什么功课？所以呢，他刚从就是Ecology 他的演讲刚有，就是说你做研究你要有一个 hypothesis， 所以基本上你怎么做是就是 under 在这个 hypothesis 的 guideline。你是借由它来指引你去怎么走下去这样，所以你们的作业呢，就是你去找一个你关心的环境的议题，然后你把它写成是你的 research statement， 会放一些范例给大家看。那 research statement 其实就是像你论文的。就是台湾看到的论文的第一章，里面 Chapter 1， 就是在讲你的 motivation， 你为什么要做这件事情，跟你最后的你觉得会看到的结果是什么？你的 objective。好，所以你选一个你感兴趣的课题，这个课题不见得是你实验室在进行的课题，就是你感兴趣的跟环境有关的课题，然后。你试着去写，就是你的 statement， 然后以所谓的 hypothesis driven， 方式去写。好，所以这是你们的作业，那你写完一样就直接上传到Ecology 的那个界面，那两个礼拜的时间给大家做这些事情。那我会放一些就是参考范例给大家，就是参考。那有问题的话再问我这样。好，所以这是你的作业这样。好，今天就到这里，下礼拜再开始从某个方面讲。我们快要进到就是我之前有跟大家预告，其实差不多期中之后呢，会开始进到所谓的比较个人